Hello, everyone. Welcome back to the next session of Contributed Talks. We have 5 talks highlighting some really cool neurocomputational modeling work here at CCN. I'm going to start off by welcoming Christina Mayer to the stage.
All right, thank you for having me. I'm excited to present our work on the neuronal encoding of attention-modulated reinforcement learning. So real-world decisions are complex because we're often deciding between options with multiple attributes. For example, some of us may want to try pizza while we're here in Boston. Each of these restaurants can be characterized along multiple attributes that include the ingredients they use, their ambiance, hours, price, among many others. Making an adaptive choice involves comparing the value of competing options. However, considering all of these attributes in this valuation poses a high cognitive load that's a challenge to both human and artificial intelligence. Humans, however, excel at this task. We make decisions such as these every day, and we hypothesize this comes about through the coordination of selective attention and value learning. By selectively attending to the relevant information, in my case as a New Yorker, of course I know it's ingredients over ambiance, we can efficiently compute and compare the value of competing options. Reinforcement learning formalizes this process of value-based decision-making as an agent that learns to maximize future success by taking actions and receiving feedback in the form of reward in a particular state or environment. Traditional RL models for behavior, however, fail to account for the fact that environments contain both relevant and irrelevant information. By augmenting this model with selective attention to relevant information, this decision process becomes both cognitively and computationally tractable. So despite robust behavioral and theoretical evidence for selective attention's role in multi-attribute RL, the neuronal processes governing it in humans remain unknown. It's been well established across species that the orbitofrontal cortex mediates value-based decision-making, and the lateral prefrontal cortex mediates top-down attentional control. So we sought to build on this robust body of work by investigating how these regions' dynamic, independent, and coordinated activities integrate selective attention and value learning in service of multi-attribute RL. To do this, we leverage invasive electrophysiology with 21 neurosurgical patients, stereotactically implanted with depth electrodes in both the OFC and the LPFC, while they complete a multi-attribute reinforcement learning task. And the task, called Gem Hunters, is a 3-armed probabilistic reward task. Stimuli are gems that vary in 2 attributes, shape and color. On each game, participants are told which of the attributes is relevant. In this case, it's shapes, and their goal is to learn which of its associated features is most likely to result in reward. In this case, it's circular gems. So we adapted this task from previous work and gamified it for use with our neurosurgical participants. And we found that our neurosurgical participants performed the task well. Shown here is performance for our neurosurgical sample compared to an online control sample. And as you can see, both groups performed comparably and well above chance. And shown here is a learning curve averaging across neurosurgical subjects, demonstrating as expected that their performance increases across trials as they learn the target feature. So to determine whether participants deploy selective attention in service of multi-attribute RL, we fit 2 RL models to participants' choice data. The first, a selective attention RL model that implements selective attention to the instructed relevant attribute during value learning. And the second, a uniform attention RL model that implements uniform attention across attributes during value learning. So I'll walk through these models now. On each trial, we assume that participants decide which of the 3 gems to select by computing their expected value as a linear combination of associated feature values, scaled by a selective attention weight which biases this valuation in favor of the instructed relevant attribute. Participants make a choice, receive feedback in the form of reward, and use this to compute a reward prediction error. And this RPE is used to update values associated with the features of the chosen stimulus. This update is scaled by a learning rate and the selective attention weight I previously mentioned. So our uniform attention model, in comparison, makes the same value learning assumptions, but implements uniform attention by omitting this selective attention weight. And the selective attention weight is a free parameter that influences value learning. So to elucidate this relationship between selective attention and value learning trajectories, I simulated one game for 2 agents with different selective attention weights. So first, for an agent with high selective attention, we see here that value assignment is concentrated within the instructed relevant attribute, shapes in this case. And in contrast, for an agent with lower selective attention, we see here that value assignment is more distributed across attributes, shapes, and colors. So we fit each model to participants' choice data using a leave one out cross-validation approach for maximum likelihood estimation. And we found, as we hypothesized, that the selective attention RL model explains participants' behavior better than the uniform attention RL model. We were curious to explore individual differences in selective attention, and to do this, we correlated participants' fitted selective attention weights with their task performance. And we found, as we hypothesized, a positive correlation between selective attention and task performance. So our model fitting approach confirmed that participants deploy selective attention in service of multi-attribute RL, gives us a subject-level metric of otherwise latent selective attention, which captures significant individual variation related to performance, and allows us to compute trial-by-trial regressors of inferred neural computations, namely expected value, to relate to oscillatory activity in our regions of interest. So across our 21 patients, we have 180 OFC electrodes and 215 LPFC electrodes. From each of these electrodes, we record local field potentials and epoch for our event of interest. For all the neural results I'm showing today, we time lock to simultaneous choice and reward outcome, which is reflected at time 0 in these figures. So shown here are voltage traces from one epoch in 2 example electrodes from our respective regions. We decompose this signal into its associated frequency bands. So shown in this bottom row is normalized power averaging across trials for the same example electrodes. And for today's analyses, we focus on high-frequency activity, or HFA, between 70 and 200 hertz. HFA is a metric of local encoding because it's correlated with spiking activity. And to investigate whether both regions encode expected value from the selective attention RL model, we ran a linear mixed effects model nested within subjects separately for each region, predicting trial-averaged HFA power using expected value as a fixed and random effect. And to control for additional sources of noise, we included reward outcome, the relevant attribute, chosen shape, and chosen color as additional fixed and random effects. So first, looking at the fixed effect of expected value predicting HFA power, we found in both regions that expected value is significantly encoded in HFA, and the negative beta coefficient here in both regions suggests that as expected value increases, HFA power in both regions decreases. And when we look at time-resolved data, this expected value-dependent difference in HFA encoding is evident at distinct time points around choice and reward outcome, which is shown at time 0 here. So this is data from one example patient averaging across LPFC electrodes on the left and OFC electrodes on the right. Next, we were curious if these value signals are biased by attention. To do this, we looked at the random effect slope from our mixed effects model, as it captures individual variation in the relationship between expected value and HFA. It also gives us a subject-level neural metric to relate to our subject-level selective attention weight. So first, looking in the LPFC, we correlated our subject-level selective attention weight with our subject-level random effect slope for the relationship between expected value and HFA power, and we found a significant negative correlation, which suggests that power changes to a greater degree as a function of expected value in participants that are more selectively attending to the relevant attribute. So to unpack this finding just a bit, on the right is the correlation between expected value and HFA power in the LPFC for one example participant who displayed high selective attention to the relevant attribute. And in comparison, here's the same correlation for another participant who displayed lower selective attention to the relevant attribute. So taken together, these results suggest that the LPFC is involved in directing attention to relevant information. And this result was region-specific. On the right is the same subject-level correlation, this time in the OFC, and we do not find a significant correlation, which suggests that the OFC maintains generalized value signals which are not modulated by attention. So to wrap up, across 21 neurosurgical participants, we found that participants deploy selective attention in service of multi-attribute RL. Selective attention correlates with task performance. OFC and LPFC high-frequency activity encode expected value from the selective attention RL model. And these value signals are selectively biased by attention in the LPFC. Please come to my poster tomorrow to hear some ideas about how these regions are functionally coordinated, or chat about the results from today. And with that, I'd like to thank my labs, especially my PIs, Angela Radulescu and Ignacio Saez. Thank you.
Great, thank you, Christina. We have a few minutes for questions. We'd like to prioritize this while trainees can come up first and ask questions at either of the mics here in the aisles. I'll start with a question. Do you have hypotheses about what's generating variance in selective attention across subjects? How can we model that? What are the sources of variation?
Yeah, so variance in selective attention, I guess it's indicative of the LPFC's potential role, as I mentioned, in guiding attention. I think something that we want to look at in terms of its utility is how this information is being shared between the OFC and the LPFC. Based on some previous work in non-human primates and ideas about corticostriatal loops, I think our hypothesis is that the LPFC might guide activity in the OFC by first maybe deciding what's relevant or sending some kind of top-down signal of the representation, which the OFC then uses to learn effectively.
That's basically what I was going to ask. I was going to ask how you think the two regions are functionally connected, but since you kind of already touched on that, what would you do to test how they're functionally connected next? Like, what is your next step?
So I think our next steps is looking at lower frequencies with the same analytical framework. So in particular, a beta coherence between the regions we think might be a mechanism of top-down attentional control. Also, theta is implicated in its attention sampling. And then in terms of directionality, we can look at metrics like Granger causality to test that hypothesis that the LPFC is in fact guiding the OFC. Thank you.
All right. Let's thank Christina again for a great talk. Okay, next we'll have Davide Geza talk about control adaptation through selective suppression of multidimensional distractors. Take it away.
All right, so crowded poster session might be familiar. To effectively follow a poster presentation during a poster session like this, we need to manage multiple sources of interference. However, how exactly our cognitive system orchestrates attention to many sources of information is not very well investigated. And this is the direction that we took with this line of work. What we found is that in humans, attention can be highly selective. And for instance, we're going to prove that people can simultaneously increase and decrease their gain for information processing on different sources of information in one-shot adaptation. This level of selectivity pushes us to reconsider traditional mode of cognitive control. So in traditional interference paradigm like a Stroop, the Simon, and the flanker, the hallmark of cognitive control is the ability of the system to relax or focus attention as a function of interference history. However, these canonical tasks usually assume that there is only one source of interference. So what we did is to increase the number of dimensions to 4 in our paradigm. These are our multi-paradigm. On any trial, you see 2 objects, left and right, that differ across 4 dimensions: the color, the shape, the edge, and the motion of the dots inside them. But on any given trial, participants are really supposed to pay attention only to one of these dimensions, as cued by the letter at the center of these 2 objects. Specifically, they are instructed that given any dimension, they have to look for a target feature. So this could be an example instruction that is given to a participant at the beginning of the task. So if you see the letter C, you need to find and respond to the blue object. If you see the letter S, you have to find the response to the oval object. If you see the letter E for edge, you have to look for a dashed border or edge. And if you see the letter M, you have to respond to the object as upward motion. So this makes up for a multidimensional task interference paradigm with a parametric level of congruency. What do I mean? Any dimension that is not queued on a given trial can act as a distractor, and it can prime the correct or incorrect response. So therefore, we have this parametric level of congruency, and this that is expressed on an x-axis here. So 0 to 3 is represent a number of non-Q dimension that is congruent with the Q dimension. And this congruency clearly predicts the response time and accuracy. For the sake of time, I'm going to focus on response time, but all our results replicate when we consider accuracy. Each dimension, when it's not queued, so when it's a distractor, has some level of interference power. And the potency of this interference depends on the dimension, and it will be relevant for later. But here, we want to talk about adaptation, so what happens on a trial-to-trial basis as a function of interference history. So in our multidimensional design, we can look at these. So we can look at the interference effect, as we've been doing before, on an x-axis, and we can look at how it is moderated by the level of congruency in the previous trial. So you can see that the slope of this line is moderated by the congruency level in the previous trial. For instance, the flatter line corresponds to a condition in which a trial followed a trial in which 0 non-Q dimensions were congruent. So a high level of interference that induced an increase in attentional focus and robustness against new interference in the following trial. But with this design, we can also have a more granular look at these adaptation effects, and we can look at the interference of each dimension as a distractor on a y-axis as a function of whether the same dimension or a different dimension was congruent versus incongruent in the previous trial on the x-axis. You can observe that only on-diagonal cells show a positive number, and this is evidence for dimension specificity in attentional control. We can simplify this heat map looking at like collapsing across off-diagonal, and again, this means that the change in interference produced by any given dimension is only function of the history of congruency of that one dimension and not any other dimension. So within dimension sequences create adaptation, but not across dimension sequences. So how can we model this? Canonical paradigm and canonical adaptation has been successfully modeled by this 23-year-old connectionist model proposed by Bottomley and Conlick. Basically, they proposed a conflict control loop that measure response conflict and use it to adjust attention on the following trial. And this works well when you have a two-dimensional task like the flanker or stroop, but it doesn't really work with a multi in simulating our kind of adaptation, because we have seen that our adaptation in our task really track the source of interference. So we adapted this model, first of all, increasing the number of pathways. So these perceptual pathways now track each dimension. And we use the diffusion process to simulate the response time. But really, the interesting addition is moving the level of monitoring from the response level to the representational level. Specifically, we measure conflict as off-field energy within each pathway as the coactivation of intermediate and response unit weighted by their connection. Now, because of the architecture of the model, this simple solution allows us to reproduce both parametric congruency sequence effect and dimension specificity in this adaptation. Furthermore, at a theoretical level, I think that thinking of a conflict in these terms allows us to move from a univariate conflict signal to a multivariate representation of conflict. And this has interesting implication in how we can try to solve the multidimensional conflict adaptation problem at the neural level. And this aligns well with the recent evidence that the anterior cingulate cortex that is implied in conflict detection, in fact, can integrate representation of a distributed representation of task variables. So in the second part of the talk, I will want to quickly cover an EEG study that we developed after these results. We have a clear neural prediction. Conflict history should selectively affect the neural representation of distractors and not the representation of the target feature. So how can we measure attention to distractors, given that distractors change on a trial basis because the task change and the cue rotates over trials? So I cannot really go into details about the methods, but I want to give you the gist. We expanded a two-step technique following the work of Tsushiki Kumoto. Basically, it consists of a first step of EEG classification, in which we try to classify what is the identity of the cued feature that people are supposed to look for and respond to. And a second step in which we use this, the classification profile that we obtain from this first step, and we regress it onto relevant model vectors. So EG plus RSA. So we can obtain a time course of relevant representation, like a representation of the task rules, representation of the spatial attention in yellow, and a representation of both cued and non-cued target features. And this is relevant for us because we can use the representation of non-task, non-cued features as an estimate of attention towards distractors. Here is represented in this black signal. Again, I have to glide over the methods, but we can decompose this distractor representation into the single distractor. And I think it's interesting to see that the strength of this representation of the distractors maps well with the potency of each dimension as a distractor, with pattern being the weakest in this task. By the way, pattern replaced motion in the EEG study. Okay, what about our prediction? We predicted specificity in adaptation, and we can, if you remember the heat map from the simulation, we can now use the same approach to analyze the EEG data. So we can align the distractor representation on a y-axis, and on an x-axis we contrast whether the same or a different dimension were congruent in a previous trial. So we can obtain a time course of adaptation. And as you can see, adaptation over diagonal emerges around 300 milliseconds, peaking around 400 milliseconds, while adaptation off diagonal is completely absent. And this is direct confirmation of our prediction that adaptation in our task is highly dimension specific. We can again break down this signal by the constituent distractors. And again, we see that the potency, so the strength of this adaptation at the neural level maps on the strength of adaptation at the behavioral level. So I've shown you that when facing a more complex and multidimensional environment, people can exhibit a high level of specificity in attentional control with this simultaneous adjustment of attention to different sources. And we can only capture this by assuming multiple parallel conflict control loops. And we have behavioral neural evidence confirming these predictions. So if you want to know more about the modeling, you can check out our preprint. And if you want to know more about the EEG work, you're very welcome to come and chat tomorrow in our poster session in the morning. I want to thank my lab, my PI Wouter Kohl, and my lab manager Tia Zalabak. And a special thank you note to Mike Freund and Tsushiki Komoto for great conversation and their help in developing the EEG methods. And thank you all for your attention.
All right, thank you, Davide. Unfortunately, we don't have time for questions, but you can feel free to reach out to Davide in the social after this session or tomorrow at the conference. Thank you. Next, we have Sang-Ho Lee, who will tell us about inverse reinforcement learning and how it captures value representations in the reward circuit in a real-time driving task. Welcome, Sang-Ho.
Okay, hello everyone. Thank you very much for attending the presentation. My name is Sang-Ho Lee from Seoul National University. I'll begin by introducing our recent paper to give you the background for the current approach. The topic of our study was impulsivity, which is a personality trait often associated with maladaptive behaviors such as drug addiction and reckless driving. Traditionally, we measure impulsivity using very simple tasks where participants choose between two options. A problem is that behaviors in such tasks are too simple to reflect impulsive behaviors in real life. To address this problem, we developed a new task named the highway task, which resembles the driving situations in real life. Let's see the performance example of a participant. Participants should control the green car to drive as fast as possible without crashing into the yellow cars. They can increase the speed to gain more scores per second, and the episode is over when the fuel becomes zero or the car crashes into a yellow car. So now the episode is over. Our goal was to model the individual differences in the behavior that reflect impulsivity using a computational model. We pose this problem as an inverse reinforcement learning problem. IRL is a method to infer the reward function of each participant by observing the behaviors in the task. The algorithm we use utilized deep neural networks to approximate the reward function. So once trained, the neural networks could calculate the reward for any possible state in the task. The main assumption here was that the reward function would reflect the subjective rewards of each participant. So in this framework, we can train a model that represents each participant in that experiment instead of training a single optimal model as in a reinforcement learning approach. To briefly summarize the results of the study, the model trained by the IRL successfully predicted the behaviors in the task and provided indicators of impulsivity. The graph here shows the proportions of the 5 possible actions in the task, turning left and right, doing nothing, acceleration, and deceleration. The blue bars show the proportions of these actions in the actual data, and the orange bars shows the model prediction. The model prediction closely resembled the data, suggesting that the IRL learned the behaviors pretty well in the highway task. An interesting analysis we can do with the reward functions from the IRL is a reward trajectory analysis, where we investigate the real-time changes in the reward during the specific events in the task. Our main finding was that the reward at risky situations correlated with self-reported impulsivity of the participants. A good example is a situation where the participant overtake a preceding car. The graph on the top shows the changes in the reward during the overtaking event. The orange line is from participants with high self-reported impulsivity, and blue line is from participants with low self-reported impulsivity, and the vertical line indicates the onset of overtaking. You can see that the reward is generally higher for the impulsive participants at around the onset of overtaking, suggesting that they prefer this risky but rewarding action. So the results show that the IRL can be a useful framework for modeling individual differences in a real-time task. So here comes our research question. Recall that we assumed IRL infers subjective rewards of the participant. This leads to another assumption that the rewards inferred by IRL would reflect the reward processing in the brain. So our current study addresses this problem. We conducted a fMRI experiment using the highway task. Our abstract contains the results with 10 participants, but we have collected more than 30 so far. In the analysis, we focused on overtaking and crashing events because these were the main events of interest in our past study. We first analyzed if overtaking and crashing triggers the ball signal in the reward circuit using a generalized linear model with onsets of overtaking and crashing as independent variables. The results show increased activation of the striatum and decreased activation of the insula during overtaking events, and the opposite patterns in the crashing events. The striatum is often associated with reward processing, whereas the insula is often linked with negative emotion processing. That is, overtaking increased the ball signal related to reward processing, whereas the crashing did the opposite. This is reasonable because overtaking is a rewarding event, whereas crashing is a punishing event. So these results suggest that the events in the high-weight task strongly affected the reward circuit in the brain. We then conducted a time series analysis to see if the ball signals in the reward circuit correlate with the IRL rewards. For this analysis, we generated the trajectories of the ball signal for 50 ROIs specified by Neuroscience. In a penalized regression analysis, we predicted the rewards from the IRL using the trajectories of the ball signal from 50 ROIs. By obtaining the beta coefficients for each variables, we could assess the contribution of each ROI in predicting the IRL rewards. The images here show the map of beta coefficients for ROIs with significantly high contributions. You might have realized that the map resembles the regions activated in the GLM analysis in the previous slide. In general, ROIs related to reward processing predicted the IRL rewards. As an example, I'll show you the trajectories of the ball signal and reward trajectory in overtaking events. This graph shows the standardized ball signal and standardized IRL reward adjusted by the hemodynamic response function. An ROI within the striatum showed pretty high correlation with the reward trajectory. And another region of interest, insula, showed surprisingly high correlation with the reward trajectory. So these supported the hypothesis that the rewards inferred by IRL reflect the reward processing in the brain. We also identified a region that provide indicators of impulsivity. The trajectory of the ball signal from the DACC resembled the reward trajectory from the behavioral data. The higher the impulsivity, the larger the ball signal from the DACC at around the onset of overtaking. This again supported the similarities between the ball signal and the IRL reward. So to conclude, we show that the IRL can be a useful framework for modeling individual differences in both behaviors and reward processing in the brain. If you want to hear more about the study or talk about potential collaboration, please come see me at the poster tomorrow. Thank you for listening.
Great, thank you very much, Sang-ho. We have a few minutes for questions. I'll start with the first question while we're thinking. Did you find either by looking at more of the behavior or in the downstream neural analysis other sort of events of interest other than overtaking and crashing that were strong positive or negative rewards?
Yeah, we actually consider other events like overtaking without lane changing or actually not that many other than overtaking and crashing. Yeah, because the goal of the task was to overtake as many cars as possible within limited time without crashing. So overtaking and crashing were two most salient events. So it was pretty difficult to specify salient events in the task other than them. But I can think of other events that can be analyzed. I'm looking for different ideas on the task.
Cool. Well, it's a really rich task. Great job. Go ahead, Christina.
Thanks, great talk. I was wondering, I don't know if this is a direct inverse, but I was thinking something more like procrastination being the opposite of impulsivity. If you picked up on, if your model is sensitive to those kind of behaviors, and or if you looked at that in the neural signal. So maybe a dampening in the regions that you sort of be activated in line with impulsivity.
Somehow it is pretty hard to hear the questions.
Maybe I'll...
Should I step back?
Just if you notice anything, I guess the inverse of impulsivity, maybe being procrastination or like failure to start or engage in actions, if you looked at that with your model or with your neural results.
Maybe the models are sensitive to traits other than impulsivity, but we haven't looked at other traits actually, but we are analyzing the correlation with compulsivity and other clinical traits that can be explained by the models. So that will be our future topics of research.
Thank you.
Mm-hmm.
One more question. Go ahead.
Thanks for the great talk. I was just curious how these effects scale with difficulty. So is there a stronger effect of reward when the car is moving faster, so the task is more difficult, or there are more cars on the road?
Yeah. Well, I think the task difficulty may or may not affect the magnitude of the reward, because the reward is always relative. I feel that impulsive participants feel the task more difficult than less impulsive participants. In overtaking events, they show higher reward. But so maybe people who feel the task difficult show higher rewards for challenging situations more than people who feel that the task is easy.
Thank you.
Thank you.
All right, thank you again, Sang-ho. Okay, up next, please introduce Jacob Tanner, who will tell us about modular boundaries in recurrent neural networks. Take it away.
Hi, everybody. Thank you for coming today. The title of my talk is Modular Boundaries in Recurrent Neural Networks. And I'd like to start this talk with a rhetorical, maybe somewhat provocative question. How can we lesion a neural manifold? Can we causally manipulate individual dimensions of this lower dimensional space to understand neural systems? I think this is an important question to ask because I find dynamics in low-dimensional spaces to be one of the most important, powerful, and intuitive tools for understanding the connection between brain activity and cognition. But the question for me is, at least with linear methods, are we stuck with abstractions like these when we're thinking about low-dimensional dynamics? The primary reason why I think the idea of lesioning manifolds is somewhat paradoxical involves the way that manifolds are sometimes defined. For example, if you use principal component analysis, or PCA, to find new sets of dimensions for neural data, you'll notice that it puts continuously valued weights on each neuron in association with each principal component. What this means is that every neuron contributes to every dimension, and therefore a lesion to any neuron is in some real sense a lesion to every dimension to one degree or another. Now, PCA uses an object called the covariance matrix to find these lower dimensional dynamics. So for example, we can grab any two neurons' activity time series, calculate their covariance, and can continue to do this for all pairs until we fill out this full covariance matrix for the system. PCA will then perform eigendecomposition on this matrix and reorder the eigenvalues and the eigenvectors. But this isn't the only thing that you can do with a covariance matrix to explore lower dimensional dynamics in a system. You can also use a network science method called modularity maximization on the standardized version of the same matrix, the correlation matrix. So what modularity maximization does in its most basic form is to reorder the rows and the columns of this correlation matrix until it finds a set of modules where there's more positive correlation within modules than between modules. And as you can see, the trajectories yielded by PCA and modularity maximization are highly similar. But whereas principal component activation is somewhat abstract and can be somewhat difficult to imagine, module activation is clear. Each axis in module state space represents a different set of neurons firing. The boundary between modules is one of the most important differences between these two methods for exploring lower dimensional dynamics, given that no such boundary exists with PCA. If we lesion a neuron, we simultaneously lesion all dimensions of the manifold. And in this way, this is with the problem I pointed out earlier. But because of these boundaries between modules, we can separately lesion neurons within one module or within another module. So we can ask this question: do the boundaries between these modules matter to the system? If they do, perhaps this is evidence in favor of a modular hypothesis about neural systems, evidence that these systems, at least under certain conditions, are partially decomposable into modules. This would also allow us to explore lower dimensional dynamics in a less abstract manner, dealing now with separate groups of neurons rather than a manifold. To ask this question, we used recurrent neural networks, or RNNs, and we trained these RNNs on systems neuroscience tasks using a toolbox called NeuroGym. This is an example of a perceptual decision-making task from that toolbox. Here, the RNN's job is to decide which of these two distributions has the greater mean. Importantly, the RNN receives inputs from sampled from these two distributions across time. So to solve this task, it needs to track the cumulative difference between the means of these distributions. With a little bit of information at time point 1, the RNN might estimate that the first distribution has the greater mean. But over time, as more information comes in, by time point 10, this estimate changes in favor of stimuli 2. And the RNN becomes more and more confident in this by time point 20. And so across time, the RNN needs to track this latent variable, namely the current estimate of the difference between the means of the distributions, what we call cumulative delta inputs. After we trained the RNNs on this task, we found that the correlation matrix of the recurrent neuron's activity did exhibit modules. You can see them in this block-like structure along the diagonal. Now, if you look at these plots on the right, you can see how the mean activity of these modules relates to the relevant latent variable for this task. Population 1 activates when cumulative delta inputs is positive, and the estimate is in favor of the first distribution, whereas neuropopulation 2 activates when cumulative delta inputs is negative, and the estimate is in favor of the second distribution. This suggests that these two populations track separate task conditions. But because these are modules, we can use lesions to investigate if this separation, if these boundaries, matter to the system. Indeed, when we lesion all connections from neuropopulation 1 to the output neurons where the decision for the RNN is made, so not allowing the system to use that information, it is only performance on one condition that is affected, whereas lesions to the other population only affect the other task condition. In other words, in this task at least, the boundaries between neural populations matter for the selectivity and the representational content of different neurons. But what about neural dynamics? Do these boundaries matter for the system's neural dynamics? We ask this question by perturbing the RNN. For example, we can add artificial input to input neuron number 1. With this, the RNN will estimate that distribution 1 has the greater mean. By looking at the principal components and using fixed point approximation techniques, we can get an idea of how this information is maintained. We see many fixed points arranged into a line attractor, a dynamic object that's good for tracking continuously valued variables. Along this line attractor is a decision boundary. If the state of the system is on one side of this decision boundary at the end of a trial, then one decision will be made. On the other side of this decision boundary, a different decision will be made. With the perturbation of input neuron number 1, the activity of the system follows the line attractor up, to the left, and away from the decision boundary. With the perturbation of input neuron number 2, the activity of the system follows the line attractor up to the right, maintaining information for either stimulus along this line. We can use this basic perturbation setup to investigate the effects of lesions to connections within either module. Lesion within population 1 results in the endpoints of stimulus 1 perturbations decaying to the decision boundary, as you can see here. whereas the information can still be maintained for stimulus 2 perturbations. When we lesion connections within module 2, however, the opposite takes place. We find this basic pattern across 100 separately trained RNNs. This, again, suggests that the boundaries between modules that are imposed by this modularity method actually matter to the system, at least in this task. Overall, this suggests that neuroscientific studies could benefit from using both modularity and manifold-based methods to understand their data and its relationship to dynamics, cognition, and behavior. That's the end of my talk. I'd like to thank my collaborators, especially my supervisor, Rick Betzel, for his support over these years. And if you're interested in talking in more detail about these results or hearing about our other results, including insights into the structural origins of these boundaries, empirical validation of some of this, and some more stuff on the dynamics of these networks and these boundaries, please come see my poster tomorrow at 11:15 a.m. Thanks.
All right, great work, Jacob. Thank you. We have several minutes for questions.
Hey, yeah, that was an awesome talk. Thanks a lot.
Thanks.
I'm wondering what the relationship this is, what relationship this has to like classic factor rotation methods, where you try and rotate your basis so that you load things onto one factor or another. And it seems kind of surprising that you would have a manifold and then rotate it, and then it's not a manifold anymore.
I am now understanding why people can't hear from this position. It was very strange on the other side.
Factor rotation methods from
Okay.
classic psychometrics and whether or not those are manifolds. Like if you just rotate your principal components using factor rotation methods, does it like stop becoming a manifold at some point along that rotation?
I mean, my intuition is that it wouldn't. But does the question involve the modular component at all? Like, would they?
I mean, it just seems like it's like a different loss function, right? So these factor rotation methods will have a different kind of objective function that they're trying to optimize. Instead of mean squared error, it'll be something else depending on the rotation method. And I'm wondering how that ties into the way you're thinking about this like categorical distinction between manifold approaches like used in PCA analyses and modularity-based approaches.
Yeah, that's a great question. I'll have to think about it some more, but I'd love to talk about it more at the poster tomorrow or after this.
Really cool work. Yeah, thanks.
Thank you.
We have time for another question. Come on down.
Hey, thank you for the talk. I'm wondering whether you see similar modules forming across different initialization conditions in the RNNs.
A weight initialization? Yes, and that's a supplemental figure. I'm not actually sure if it's still in the paper. But yeah, across different initializations, we find that similar modules emerge.
Great talk. I'm curious about a follow-up question related to the nature of the modules that you see. How big are your networks, and do you see any dependence on the size of the network for obtaining similar modules across initializations?
That's a great question. The networks are 100 neurons, and so they're large. But when they're smaller, there's still modules there. I think probably up until the point where maybe the number of modules that we see here is the same as the number of neurons that are in the RNN. That might be different. But I don't know. I'm not sure exactly how we would compare the modules across different sizes of neurons. I think was that your question? Sorry.
Well, you mentioned that you see similar modules reappear across initializations, and I'm wondering if that's true at all scales of the network that you train.
Sorry, what was the last part?
I wonder if that's true for both small networks and larger networks.
Yeah, I guess I'll just have to think about it more, because at least off the top of my head, I can't think of a method for comparing the similarity of the modules when the number of units is different.
Time for questions, but thank you again, Jacob, for the great talk. All right, our final speaker is Debbie Yi, who will tell us about neural computational mechanisms of motivational influences on mental effort. Take it away.
Can you guys hear me? I recognize that I am the last thing between you and coffee, so thank you for sticking around for this last talk. So I'll just go ahead and get started. So today I'll talk about a fundamental question in goal-directed decision-making, which is how do we decide when and how to allocate cognitive control? And for those unfamiliar with this term, cognitive control is the ability to flexibly process task-relevant information in the service of completing a goal. So for example, imagine that you're working on some manuscript or grant proposal, and if you're successful, you'll receive positive feedback or reward for completing your task well. But conversely, failing to reach those goals may be associated with negative outcomes, and you may receive a penalty for poor performance, may be fired, there's like negative consequences for failure. And prior work has shown that both positive and negative outcomes shape decision-making, but exactly how the brain represents diverse motivational signals to drive control allocation remains unclear. And so one neurocomputational framework that has been useful for addressing this question is the expected value of control framework. And according to the EVC, individuals can exert varying levels of cognitive control when performing a task. And depending on the efficacy of their actions, as well as how much control they exert, this can result in either good or bad task performance, leading to either positive outcomes, such as a monetary gain, or negative outcomes, such as a monetary loss. And so a key feature of the EVC model is that the amount of cognitive control that we choose to allocate on a given task is determined by the net difference between the expected benefits, which we here define as the combined value of the positive and negative outcomes, and the expected costs, which is defined as the effort cost required to exert a certain amount of cognitive control. And so importantly, a key hypothesis arising from this framework is that humans will integrate the value of positive and negative outcomes into a motivational value signal. And then this signal will subsequently be translated and drive cognitive control allocation. And so to formally test this hypothesis, we developed an incentivized self-paced Stroop task where a color word is displayed and participants had to respond to the ink color. And importantly, because participants can complete or as many as few trials as they choose within a fixed time interval, this allows us to quantify how much effort people choose to invest over a period of time. And prior to each interval, participants see a cue that indicates the type and magnitude of potential outcomes for correct and incorrect responses. And at the end, participants are shown their cumulative feedback after each interval. And so in this task, participants could earn high or low monetary rewards for correct responses and were penalized with high or low monetary losses for incorrect responses. And so recent work from our group has shown that positive and negative incentives guide dissociable strategies for cognitive control, with the assumption that individuals are maximizing their expected reward rate. And so here, I've just listed the normative reward rate model, which formalizes expected value as the integrated value of expected rewards for correct responses and expected penalties for incorrect responses, normalized by response time and subtracting and regularized by an effort cost. And I won't have time to go into detail today, but I just wanted to briefly mention that we use drift diffusion modeling as our underlying process model. And so based on this model, we hypothesized that participants would optimize their performance by choosing a combination of drift rate and threshold, which are key parameters of the DDM, to maximize their expected reward rate. And so critically, our model simulations predict that a larger reward would encourage participants to increase their attentional focus on the Stroop stimulus, which would be reflected as an increase in drift rate and reduced threshold. And conversely, the model also predicts that larger penalty would encourage participants to respond more cautiously to avoid making mistakes, which would be reflected in an increased threshold. And so we've run several studies with this task and this model, and consistent with our normative model predictions, we found that rewards and penalties influence dissociable control strategies with individuals exerting more attentional control when they're anticipating monetary gain versus more caution when they're anticipating higher monetary loss. And so this begs the question, what are the neural mechanisms underlying this control strategy? And so to empirically test this question, we conducted an fMRI study in which participants performed the multi-incentive control task. This allows us to acquire a more granular understanding of the neural circuitry underlying the interaction between motivational incentives and cognitive control. And so while there's a lot of questions that our study can address, today I'll focus on results that address these two. First, to what extent do brain networks represent control strategies for attentional control versus response caution? And second, to what extent do expected reward and penalty incentives influence these control strategies? And so here, I just focus on a set of a priori ROIs based on the literature. And so we selected brain regions that encoded the subjective valuation of incentives, such as the ventral striatum and the anterior insula, as well as motivated cognitive control, such as the dorsal anterior cingulate cortex and the lateral prefrontal cortex. And we extracted cue-related activity during the task and included these neural regressors as covariates in our drift diffusion model. And in particular, we used two subnetworks of DACC based on their categorization in functional networks. Here, we just label as caudal and rostral DACC, respectively. And so consistent with previous behavioral studies, we replicate the main effect of incentives on control strategies consistent with our normative predictions, with reward predicting increased drift and reduced threshold, and conversely, with penalties predicting increased threshold. And intriguingly, when we look at the brain data, we observe an interesting pattern through which these regions are predicting the main effects of our two control strategies, with the caudal DACC associated with increased drift rate, and the rostral DACC is associated with increased threshold. But what about the effects of incentives? And so across several of our ROIs, we observe an intriguing pattern consistent with our normative predictions, with rewards interacting with ROIs to reduce their response threshold, and penalties interacting with ROIs to increase their response threshold. Oops, yeah, sorry, there's a bit of a lag. So as we can kind of see here, you just kind of see in the yellow that there's a bit of a drop and an increase here with in the threshold that is interacting with the incentives here. Okay. So just in conclusion, we find that distinct dorsal ACC networks predict the main effects of control strategies, and also interactions with our ROIs reveal that the salience of the incentives guide control allocation. However, one intriguing question that arises from this data is that it's unclear to what extent DACC activation is associated with control signals per se. So specifically, are they specifically predicting drift rate or threshold? Or rather, does this activation reflect how reward and penalty incentives are just driving variability in control allocation based upon the expected goals of the task? And so ongoing work seeks to clarify the nature of how DACC and other relevant regions represent the interaction between motivation and cognitive control. And specifically, it would be important to clarify to what extent these regions are driven by the likelihood of success or failure in task performance versus the values of the motivational incentives that are driving these dissociations and control strategies. And so if you're keen to chat more, just please visit me at my poster, C162, at tomorrow's poster session. And with that, I'll just like to thank my lab, my advisor Amitai Shenhav, and the rest of the lab without whom we wouldn't have run this exciting set of studies. So thanks for your attention.
Thanks, Debbie. We have time for a question or two. Go ahead.
Thanks for a great talk. I'm interested in your model. Are you interested in studying the update on?
You speak into the mic.
Oh, yeah. In your model of control, do you also look at the change in control after the outcome of the from the action at all, like the updating control?
Like in terms of like the trial by trial feedback?
Yeah.
Oh, okay, so yeah, so that's a good question. So the question you're asking is like, is the allocation of a control modulating the subsequent trials, like control and subsequent trials as well? So in this study, we're not looking at it. One of the limitations of this, the way that we did the hierarchical modeling is that it collapses all the trials across different conditions. There's other work in the lab. Ivan Grahek is somewhere around here. He's done some really exciting work looking at learning and how like learning from feedback modulates kind of adjustments in control. And so in this project, we haven't looked at it per se, but it is the case that people do learn how to adjust control based upon the probabilities of the expected outcomes as well as the valence of the expected outcomes.
Cool. And is there any neural findings of that as well?
Also, some of it is ongoing. There's some nice EEG work by Romy Fromer, who's also floating around here. And she has run an EEG study looking at kind of the different signals that are associated with kind of reward and the efficacy of people's actions driving control strategies.
Cool. Thank you.
We have time for one more quick question, if there is one. If not, then let's thank Debbie again for a great talk. All right, well, that concludes our session today. We'll be back in this room at 5:15 for the community event, the Battle of the Metrics. And we have a separate generative adversarial collaboration happening over in the Solid of Puerto Rico as well at 5:15. Until then, enjoy your break.